As with any living thing, our body is made up of cells. The forces between these cells, or cell junctions, are crucial to keeping our tissue intact. Cell monolayers, while only being one cell thick, not only act as the physical barrier between internal and external environments within the body, but it must also withstand a large amount of mechanical stress. For example, the innermost layer of the cornea is made up of a cell monolayer. Currently, any damage to this tissue is repaired through a donor transplant. Lab-grown monolayers could potentially eliminate the need for donor tissue. However, researchers first need to ensure that these lab-grown monolayers are able to withstand the act of transplantation, as well as the forces within the eye itself. There is a significant lack of devices able to test the cell-to-cell junctions making up the strength of a cell monolayer. It is because of this unmet need that our team has been tasked with designing a device capable of measuring the mechanical properties of a cell monolayer.
We have designed a device that is able to produce a free-floating monolayer and quantify its deformation both radially and from a side view using an applied force using phosphate buffered saline, or PBS. To accomplish this, we use a 1 centimeter tall tube of condensed polystyrene, a material which is known for its biocompatibility and hydrophobicity. We then mill a 1.75 millimeter hole through the tube, which is where our applied force will take place. Then, we pipe at around 50 microliters of gelatin into the bottom of the tube and allow it to cross-link atop a glass surface in order to keep the gelatin surface as flat as possible. A solution of PBS is then added to allow it to reach a swelling equilibrium while remaining flat, in addition to keeping the gel hydrated.
Once the gel is fully set and swelled, we will then flip our device over and seed our surface with corneal endothelial cells and media via a droplet. We allow the cells 1 to 2 days to fully adhere to the surface of the gelatin and device, and after this 1 to 2 day window, we then transfer our tube into a 24-well plate, where we will then submerge the device in cell growth media to allow for a proper flow of nutrients. Next, once our cells have reached full confluency and have formed a monolayer, we will gently place our device sideways into an enzymatic bath of collagenase for 1 hour. This step fully removes the gelatin plug that was placed within the tube and creates a free-floating monolayer across the 1.75 millimeter hole with cells attached only to the outer rim of the tube. After this, we place the tube into a bath of PBS to quickly rinse off any excess enzyme, and then into a bath of host and cell mask for 30 minutes. These two products are intended for staining the cells and their nuclei for visualization during the image analysis step.
After the cells are stained, we then place the tube into the device stand situated on top of a confocal microscope for radial view. An additional microscope is used to capture a side view. A force is applied to the cells using a syringe pump and microfluidic tubing. This force is applied until the material fails, meaning until the cell monolayer breaks. Using the images captured from both the confocal and side view microscopes, mechanical modeling is applied to the segmented images. Using these equations, we are able to produce stress-strain curves to model the mechanical behavior for the monolayer, as well as quantify its mechanical characteristics, such as yield strength, perimeter contractility, contractility modulus, and area elasticity coefficient. Using a biomaterial film stained blue for easy visualization, we were able to produce a proof of concept and confirm that this method does work. Although we encountered setbacks inhibiting us from culturing a full monolayer on our tube design this year, we were able to use a biomaterial film to complete testing of the mechanical modeling. As this project continues and successful monolayer culture has been achieved, the imaging and analysis methods can be used to quantify the mechanical properties of a cell monolayer.